Alright, you're welcome back, and let's continue to from number 16 to where we'll reach. Now, if you missed number 1 to number 15, please go back and watch number 1 to 15 because you missed a lot if you missed that one. Alright, welcome back to my YouTube channel. My name is Excellence Simplified, and I'm going to be walking you through your FSB exam. Let's go. Number 16 says the hydrolysis of fats by alkali is known as dash. The hydrolysis of fats by alkali is known as dash. The answer to this question is saponification. Saponification. Saponification is the process of making soap. Abi, we want to make soap. We want to make soap. Now, the process of making soap, you always use fat and oil. You can use oil, like there's a specific oil you use. Then you use a base. Now, that alkali hydrolysis, alkali is a base now. Alkali hydrolysis of fats is called saponification. Saponification. All right. Number 17 now. The fluidity of the plasma membrane is maintained by dash. Now, plasma membrane. When they say plasma membrane, you agree with me that we have cells. Let's say this is your cell. Now, the cell have the nucleus, right? And this open space is called the cytoplasm. It's called the cytoplasm. Now, this thing here that serves as a wall that protects that regulates what comes in or goes out of the cell is called the plasma membrane. If you don't want to call it the plasma membrane, you call it the cell membrane. So now, the fluidity is in a fluid state. This plasma membrane is in a fluid state. That fluid state is maintained. That fluidity of the plasma membrane is maintained by a substance we call cholesterol. Cholesterol. So cholesterol is the answer to that question. Cholesterol is the answer to that question. So the fluidity of the plasma membrane is maintained by cholesterol. Can we move on? Question number 18. The non-amino acid group of a conjugated protein is dash. The non-amino acid group of a conjugated protein is dash. Now what they are asking is, some proteins have an additional group that is not amino acid. Remember that we've said that amino acids are the building blocks of proteins, right? Good. Now, the non-amino acid part of the protein, the non-protein part of the protein is called the what? Can you guess? It's called prosthetic group. B is the answer there, prosthetic group. Prosthetic group is just like an additional group. Do you get? So, if you have something like glycoprotein, or let's say lipoprotein. If you have something like lipoprotein, you see this is protein, right? This is lipo, right? This lipid. So you are attaching lipid to protein. This lipid is not an amino acid. Now, this lipid here is called the prosthetic group. Prosthetic group. The lipid is called the prosthetic group. Alright, so that's that. So the answer to that question is prosthetic group. Number 19. The two strands in the double helix of a DNA, are bonded to each other by dash. Now let me draw something for you. Now, one thing you need to know about DNA is double-stranded, have two strands, and it looks like this. This is how the DNA looks like. Then this is one strand, and this is another strand. Now we have bases here. We have bases here. Now these two strands is linked together. How are they held together? How are they being held together? The two strands are joined together by a bond called hydrogen bond. Hydrogen bond. So we have bases here. The bases can be cytosine, guanine, adenine, like that. And here can be guanine, cytosine, thymine. Now there are hydrogen bonds holding these things together. There are hydrogen bonds holding them together. So the answer to that question is hydrogen bonds. So you see that hydrogen bonds are bonds that hold the double helix of DNA molecule together. Good. Can we continue? Now, number 20. Sorry, number what now? Yes, number 20. One of the following are examples of essential fatty acids. One of the following are examples of essential fatty acids. Now, what are essential fatty acids? Essential fatty acids are those fatty acids that our body cannot synthesize. So it is essential that we get it in our food. It is essential that we get it in our diet. So why is it that, have you ever asked yourself, why is it that your body can synthesize some amino acids, some fatty acids, and cannot synthesize others? Now, this is the reason. Let me show you something now. Please look up. Look up. Let me show you something. There are some fatty acids that our body can synthesize, and there are some that our body cannot synthesize. Now, any fatty acid that have double bond after carbon 9, our body cannot synthesize it. Our body will say, no, I cannot do that one. I will show you something. Any fatty acid that have double bond after carbon 9, after carbon 9, our body cannot synthesize it. Now, if you look at, let's take for example, if you have something like this. Now, if you look at this one have a total of 14, 15, 16 carbon, right? This one have a total of 16 carbon. Now, this one is called palmitic acid. Can our body synthesize this one? Yes, our body can synthesize this one. Why? Because there's no double bond set. So our body can synthesize it. So if our body can synthesize it, is it essential or non-essential? If our body can synthesize it, then it is not necessary, it is not essential that we get it in our diet. So this one is non-essential. If you have another one like this, now if you look at this one has 7, 8, 9, 10, 11. Okay, let me make it up. 7, 8, 9, 10, 11. 11. So I have 7 here. Alright, so if you check this one, I have a total carbon of 1, 7, 8, 9, 10, 11. 11 plus 7 is what? 18, right? So this one that have 18 is called what? And double bond. One double bond, if you check the double bond is on carbon 9. Because it's 7, 8, 9. Now this one is called oleic acid. Oleic acid. Can our body synthesize it? Yes, our body can synthesize it. But any fatty acid that have double bond after this carbon 9, take for example, if we have another double bond here, if you have another double bond, here is carbon 9, 10, 11. Okay, let me put it on 12. Let me put it on 12. If you have one on carbon 12 here, our body does not have what it takes to put a double bond on carbon 12, so our body cannot synthesize it. Our body cannot synthesize it. Alright, so since our body cannot synthesize it, it is essential that we get it in our diet. Now the question is asking, one of the following are examples of essential fatty acids. Now A says linoleic and linolenic acid. Now linoleic and linolenic acid, they are essential fatty acids. Our body cannot synthesize them. They have double bonds after carbon 9. B says palmitic and stearic acid. Both of them, they are non-essential. Our body can synthesize. Palmitic and arachidic acid, oleic and citric acid, all those ones are essential, are non-essential because our body can synthesize them. Now, the answer to that question number 20 is A. The answer is A. All right. Are you following? If you are following, if you've watched this video to this point, please subscribe. Please subscribe. You have many things to gain from subscribing. I post videos usually. If you go through my channel now, you see videos that will help you. See videos that will help you. So please click on the subscribe button so that anytime I post video, you gain from it. Do you understand? Because I'll be posting video. You're year 2, you see videos. By the time you get to second semester, you see videos there that will help you. Year 2, you see videos. Year 3, until you graduate, videos that will help you are on my channel. Do you understand? So please click on the subscribe button and subscribe. Let's move on. Question number 21. Which of the following scientists isolated nuclein from phosphate-rich pulses? Which of the following scientists isolated nuclein from phosphate-rich pulses? Now that the phosphate-rich pulses, this scientist isolated nuclein from phosphate-rich pulses, and the name of that scientist was Friedrich Miescher. Friedrich Miescher. Option C. Option C. So the answer is option C. You still remember this option A? This Richard Altman, what did he do? The person that introduced that word nucleic acid, abi? Yes. Now number 22. Number 22. Which of the following bonds, they made mistake there, it's supposed to be which of the following bonds are present between the sugar and phosphate groups of a DNA? Which of the following bonds are present between the sugar and phosphate group of a DNA? So what do you think is the answer to that question? What do you think is the answer to that question? The answer to that question is ester bonds. Ester. It's actually called phosphodiester bond. Phosphodiester bond. So the answer to that question is ester bond. Ester bond. Number 23. Dash wrote that nuclein is responsible for the transmission of hereditary characters. Who said nuclein is responsible for hereditary characters? The answer is A. Oscar Hartwig. Oscar Hartwig. Now number 24. The carriers of the basic units of heredity are known as dash. The carriers of the basic unit of heredity are known as dash. Now, the basic unit of inheritance. Have you ever passed on the road and they say, are you the daughter of so person? Are you the son of so person? It means you inherited something from your mom or your dad. Now, the basic unit of inheritance is the gene. The gene. So, the carrier of the basic units of heredity are known as the genes. So, gene is the answer. Gene is the answer. Number 25. The bonds that link nucleotides together are called dash. The bonds that link nucleotides together are called dash. So we've seen that nucleotides are the building blocks of nucleic acid, right? Yes. Now the bonds that link nucleotides together are called phosphodiester bonds. So the answer to that question is phosphodiester bonds. We are moving on. Question number 26. The two strands in a double helix of a DNA are bonded to each other by a dash. The two strands, we've answered this question before, right? We've answered this question before, but they put it in another way, so it's repeated now. The two strands in a double helix of a DNA are bonded together by what? Do you still remember? Hydrogen bonds. Hydrogen bonds is the answer. Hydrogen bonds. Now, number 27. An RNA contains which of the following bases? An RNA contains which of the following bases? Now, let me explain something. Now, we have nucleic acid. And there are two types of nucleic acid. The two types of nucleic acid we have are RNA. and DNA. RNA and DNA. And don't forget that the building block of nucleic acid is what? Nucleotides, right? Nucleotides. Now let's keep this aside. I'll come back to this. But let's look at this nucleotide again. There's something I need to say. There's something I need to tell you. Now, nucleic acid, we said the two types are RNA and DNA. And the building block of nucleic acid is nucleotides. And we said nucleotides are made up of two things, three things. One is nitrogenous base. Two is pentose. And three is what? Phosphate group, right? Phosphate group. Hope you still remember that if you keep the phosphate group aside, it is no longer a nucleotide but a what? Nucleoside group. Now this nitrogenous base we have here can either be a purine or a pyrimidine. It's either a purine or a pyrimidine. Now purines are two rings combined together, while pyrimidines are single rings. Examples of purines are adenine and guanine. Examples of purines are adenine and guanine. While examples of pyrimidines are cytosine, uracil, and thymine. Now, if you look at this very well, purines, adenine and guanine, the short way you can use and remember that is pure as gold. While pyrimidine, CUT, cut. So CUT, cut. Now, out of all these nitrogenous bases, this one can be found in DNA and RNA. This one can be found in DNA and RNA. This one can be found in DNA and RNA. But uracil is only found in RNA and thymine is only found in DNA. Please take note. Uracil is only found in RNA and thymine is only found in DNA. So take note of that. Uracil is only found in RNA, thymine is only found in DNA. Now, let's take a look at something. We said we have the bases, right? The bases are adenine. That's A. Guanine. That's G. Cytosine. That's C. Uracil. That's U. And thymine. That's T. Now, we said this one is only found in RNA and this one is only found in DNA. Why the other ones can be found in any of them? Now, there's what we call base pairing. Base pairing. Remember that in a DNA molecule, DNA is double-stranded, right? Now the bases, double strand. Now the bases can pair together, but the stuff here is A will only pair with T. A will only pair with T with 2 hydrogen bonds, and G will only pair with C with 3 hydrogen bonds. So adenine can only pair with thymine, 2 hydrogen bonds. Guanine will pair with cytosine with 3 hydrogen bonds. Remember that uracil is not found in DNA, so you cannot see uracil here. I'll come back to this, but let's go to the question and continue. Now, question 27 says, an RNA contains which of the following bases? A. Adenine, thymine, guanine, cytosine, uracil. B. Adenine, thymine, guanine, cytosine. C, thymine, guanine, cytosine, uracil. D, adenine, guanine, cytosine, uracil. So what do you think is the answer? You already know, right? You now, your mind will just tell you that thymine cannot be found in DNA. Remember, thymine is only in DNA, right? So thymine cannot be found in RNA. Thymine is only found in DNA. So they are asking which one is for RNA. So you just check anyone that have DNA, anyone that have thymine is out of it. A, does A have thymine? Yes. So that one is out. B, does it have thymine? Yes. So it's out. C, does it have thymine? Yes. So it's out. D does not have thymine. So D is our correct answer. D is the correct answer. Hope we understand. Now number 28. Which of the following DNA sequences is complementary? Which of the following DNA which of the following DNA sequences is complementary to 5 prime? We have TAGAC, TAGAC, then 3 prime. Now, anytime you see a question like this, the first thing you do is to smile because it's a bonus question. Remember that adenine will always pair with what? Thymine, right? And guanine will always pair with what? Cytosine. Now, if you want to write the complement, the complementary base of this, the complementary pair, the complement will always run in this direction. It will always, if you see, this one is called a template. This one we're giving is called the template. And it's starting from 5 prime and ending with 3 prime. If you want to write the complement, all you need to do, you will start with 3 prime and end with 5 prime. So the complement is always starting with 3 prime and ending with 5 prime. Now, T will pair with what? A, right? So as you see T here, you write A here. You are just writing what they will pair with. A will pair with what? T, you write it. G will pair with what? C, you write it. A will pair with what? A will pair with what? T, you write it. C will pair with what? G, you write it. So this one we have here now is a complement. You now check your options and look for where you see this one. And you click it and you go on. So let's check. Where do we see that? G, A, T, C, T, G. A, T, C, T, G. So which of the following DNA sequences is complementary to A, T, C, T, G? Alright, so you can see there option B. Option B is the correct answer. And it's running from 3 prime to 5 prime. Don't forget that. So option B is our correct answer. Alright, we are moving on, please. Why have you not subscribed? Why Nigerians? Why have you not subscribed? Nigerians, please subscribe. And if you are following, please drop it in the comment section. I am following. If you are following, drop it in the comment section. I am following. I am following. I am following. I am following. Alright, let's move on. Number 29. The double helix structure of the DNA was composed by dash. The double helix structure of DNA, this double helix, to show that it's double helix like this. Like this. The double helix of the double helix structure of the DNA was composed by, the answer is Watson and Crick. So C is the answer there, Watson and Crick. C is the answer, Watson and Crick. Alright. Now, number 30. Which of the following proteins compacts and organizes DNA in chromosomes? Which of the following proteins compact and organizes DNA in chromosomes? The answer there is histone. Histone protein. Histone protein is what organizes and compacts DNA. Alright, so this is number 30 now. If you've watched to this point and you've learned something, please click on the subscribe button. Should I continue to the other ones? Yes, I will continue. But let me know in the comment section. If I should continue, just drop it in the comment section. Okay, if you want me to continue, drop this in the comment section. I have subscribed. Continue. Drop this in the comment section and I will continue. Drop it in the comment section, I will continue. So when I will see this, enough of this, it will give me that energy to now continue. So drop this in the comment section. I have subscribed. I have subscribed. Continue. All right. So drop that in the comment section, and we'll see in the next episode. This is part 2. We'll see in part 3. All right. See you in part 3. My name is Excellence Simplified.